Hello viewers, I'm Arpita Banerjee and welcome to my YouTube channel where I discuss bioinformatics and computational biology. In this particular video, I will talk about parsing files from ClinVar. ClinVar is a database that stores information on mutated genes which cause diseases. I will talk about parsing the files from ClinVar to glean information as per my requirement. ClinVar is a database maintained by National Institute of Health. It is actually a database of genetic variations that causes human diseases. In the About tab, it would exactly tell you what is ClinVar. It is a freely accessible public archive of reports of human variations classified for diseases and drug responses with supportive evidence. Say, for example, if I type in a protein, GLUT1, and search for it in the database, here are the search results where, see here, it says with copy number gain, the condition is pathogenic for this particular sample. Clicking on any variant would give you the exact variation ID inside. For copy number loss, the classification or the sample has proved to be pathogenic for this particular sample. If we go further down, you'd find that for single nucleotide variant, it's uncertain significance, so they are not sure whether the mutation is at all of any relevance for this specific sample. Further down, you may come across cases of single nucleotide variation where the classification is pathogenic. So the sample has been shown to be pathogenic. ClinVar has hordes of information about this gene GLUT1 and the variations in the gene that causes abnormalities. You could find information on other diseased genes as well in the database. Now, if you see, there's this column called condition where they tell you the clinical condition that results out of the mutations. In case of GLUT1 gene, in most variants, the clinical condition or the syndrome is hyperinsulinism, hyperammonemia syndrome. What is hyperinsulinism? What is this syndrome all about? Reportedly, it is a syndrome which is second most common form of congenital hyperinsulinism. Children affected by the syndrome have both fasting and protein sensitive hypoglycemia. Mutations in the gene GLUT1 is causing this syndrome. And what is GLUT1? GLUT1 codes for a protein called glutamate dehydrogenase 1. It's the gene is there in mitochondria. That's why you have this mitochondrial here. The function of the protein coded by this gene is to catalyze the conversion of L-glutamate into alpha-ketoglutarate. Okay, so it plays a key role in glutamine metabolism. For all the various samples of one gene, which is GLUT1 in this case, the gene or protein change, the type of change, the diseased condition that arises out of the change or mutation, and the pathological significance are all listed here side by side in tabular format. How to get information of all the variants in one single file? You can download. See, it says download 296 items, means there are 296 variants that are, there in the database. You can download the tabular text. You can sort by gene, relevance, location. Location means location on the chromosome. As per your requirement, you may want to sort and then you download, create file. The downloaded file is named ClinVar_result.txt. That's the default name. Okay, so for whatever gene you download the file, it would be named something like this, regardless of the gene name, unless you rename it, of course. If we open the file to take a look at it, this is how the file looks. Okay, so everything in one file in text format. When I downloaded the file, I just went with the default option, that is sort by location. If you notice here, the location has been sorted. For example, in number 15, you have this 87050,203. At number 16, you have 87050,206. And then the next one is 87050,236. You see the numbers arranged in increasing order. I divided the parsing work into two sections. In one of the sections, I was dealing with the mutations and the corresponding condition, that is, the diseased condition. And in another section, I was dealing with the mutation and the clinical significance, meaning whether the mutation was benign, whether it was of any relevance at all, whether it was pathogenic. The purpose was to correlate and to derive some information for some work that I had planned at that point of time. For both the sections I mentioned, there are essentially three steps involved. Step 1 extracts entries that can be processed. You will see in a while what I mean by that. Step 2 is about parsing the entries in the extracted file for comprehensibility. If you scrutinize any downloaded file from ClinVar, you would see that it's not very readable. Of course, you can read the letters and the words, but what I mean is that the information in the file is not very clearly laid out. Therefore, to derive the gist of information, parsing was essential. Step 3 was about counting the numbers from the gist. As you see this output on the screen, you would see that step 1 is common to both the sections of correlations that I mentioned earlier. One of the sections is about correlating the pathological condition with protein change. For that, step 1 involved taking the downloaded file from ClinVar as input and processing it to extract those entries which can be further processed. I designed step 1 in a way such that my flow of codes would work for any gene. For that, the gene or the protein name had to be specified inside the code. That is to say that every time I chose to work on a new gene, I had to type in the gene name inside step1 code.py and save it before running the code. Say, for example, in this particular case, GLUD1 was specified as the gene inside the code, inside step1 code.py. If I were to work with a gene called ADK, I'd have to replace GLUD1 with ADK at a particular line of code inside step1 code.py. Step 2 was about taking the input from step 1 and running a code, running my code on it to generate a file that would have parsed information for clarity. By the way, all the codes involved in all the steps are mine. Step 3 involved taking the parsed information as input and then counting the numbers, counting relevant numbers to be churned on screen. Other section is about correlating clinical significance with protein change. Step 1 remains the same, extracting those entries from the downloaded file that can be processed. Step 2's input remains the same, that is the extracted file. However, the code has been designed such that it would output the parsed information in a way that would help the correlation of clinical significance and protein change. Step 3 is again counting numbers in the parsed output from step 2. I had a particular project in mind at that point of time that I intended to put up for funding. Nonetheless, let's start. We are in the test directory, and what do we have here? We have two subdirectories, ClinProt and ConProt for the two separate sections of correlations that I mentioned earlier. Okay, what else do we have here? The downloaded file from ClinVar. So this is a direct downloaded file. And we have step1 code.py. So this is the code which takes the ClinVar underscore result.txt, the downloaded file from ClinVar as input, and processes it. What does it process? How does it process it? We'll see it in a while. Let's run this code, step1 code.py. Okay, what do we have? We got this additional file. gene extracted underscore step1 output.txt. Now if I open this, this is what it is. How does it differ from the downloaded file? Downloaded file is this one, right? Now if you see in the downloaded file, some of the entries or some of the samples or serial numbers have a lot of genes, but we are concerned about GLUD1. If you see further down, the tendency of having multiple gene entries disappears. There's one gene which is of our concern here, which is GLUD1. Same for 17. Okay, same goes for 18 and so on and so forth. Switching back to the output file from step 1, this output file has essentially got rid of entries with multiple gene information. That is how the code had been written in the first place, to extract such output. This gene extracted underscore step1 output.ext is going to be the file which would be further processed for the correlation. Let me just copy it to conprot and copy it to ClinVar. Okay, so here we are in the conprot directory. And what do we have here? We have the code for step 2 and we have the code for step 3. And this is the file which would be inputted for step 2 code to work on. This gene extracted underscore step1 output.txt is going to be the input file for this code, conprot step 2 code.py. Okay, so if I just run the code, step 2, okay, nothing on screen, but let's see what we got here. We have this conprot parsing underscore step 2 output.txt. This is the file which is being outputted by this code over here. Okay, now if I show you the output from step 2 in this section of correlation, this is how the parsed file looks like, the output from step 2, where the information of relevance has been laid out side by side for clarity or better readability. And now this one, the output, is going to be the input for this code, the step 3 code over here. The codes have the input specified. So when I'm saying that this particular file is going to be the input for this particular code, I'm not showing the code, but inside the code, it is specifically mentioned that the input is this one over here. I'm not going to show you the code and explain, line by line. You won't find such codes out there in the internet. Now let's run the code and see what happens. Step 3. There. So this is the output I got. Conditions with no protein change is, you have the numbers over here. Conditions with protein change, where the specific change at the amino acid level is mentioned. So these are the numbers for that. Hyperinsulinism is 1, hyperinsulinism, hyperammonia syndrome is 30, monogenic diabetes is 1. Now, in case you're wondering, what is this no protein change and what is this protein change? For that, I will just show you the input for step 2. This is the input file that went into the step 2 processing code, which ultimately gave you the numbers churned on the screen. Consider this one, serial number 112. In this case, this is the change at the DNA level, which is happening. So this 228 cytosine is getting mutated to thymine. But if you see, there's no protein change which is mentioned over here. On the contrary, if you go to the next one, this is the change which is there at the gene level, and You have this arginine which is getting mutated. So at the protein level, what's happening is arginine is getting mutated to cysteine. If you look at this particular serial number, see, here's the change at gene level, which is mentioned over here. This guanine is getting converted to cytosine. So this particular mutation has the potential to affect change at position number 50, the amino acid number 50, which is arginine. But here, nothing is changing. And you see the protein change field is blank. It is a silent mutation. On the contrary, if you look at 118, there's this change at gene level. Guanine at 136 position is getting converted to or getting mutated to adenine. And at the protein level, alanine 46 is getting mutated to threonine. So here you have the protein change field, which is A46T. Alanine 46 is getting converted to threonine. Now we are in the ClinProt directory. And what do we have here? Gene extracted underscore step1 output.txt. We have the code for running step 2 and we have the code for running step 3. Okay. For the correlation between protein change and clinical significance. Running step 2, which takes this file as an input, running step 2, we got this as output. ClinVar parsing underscore step2 output.txt. You may want to take a look at the output from step 2, the parsed information, where the protein change and the corresponding clinical significance are laid out side by side for clarity. This is going to be the input for the step 3 code. Let's run the step 3 code. There. Okay, what do we have here? Total of no protein change cases, total of protein change cases, and most importantly, clinical cases with no protein change. We have the numbers here where I have actually demarcated whether the case is benign, likely benign, or a mix of two. And then going to clinical cases with protein change, and numbers are here. Okay, these numbers could matter, but I was mostly concerned with these numbers over here. I never showed the code to anyone, so this is the first time I'm actually showing the output here in this video. Not only just parsing, the codes also involved counting, relevant counting for correlating the mutations with the condition, and for correlating the mutations with clinical significance. I needed the numbers for some work that I had planned. Like I mentioned earlier, my code was designed to work for any gene. So these are some directories or folders where I downloaded the corresponding genes files from ClinVar. And as you can see from the long list, I was working with these files in 2021. Another part of the project involved deriving a pattern from studying many genes. There was a lot of coding involved in the whole thing I did. Lot of coding. It took me months for writing all the lines of code. And you just saw it in one click and whoosh, you get the result. But it's not that easy as it looks. For my purpose, I used this book, Learning Python by Mark Lutz. I bought this book in 2019. When I had read the book for the first time in 2010, it was this thick. And now it's this thick, almost 4 times the volume. So for my coding work, I used lessons from this book as well as quick look up a few commands from the internet. It took me a lot of effort, time, and patience. I had started the coding in 2020. I continued for a few months at a stretch. Then there was a disruption. because of my computer issues and stuff. I transferred all my files, all my coding files, etc., into my new computer in 2021, and I continued working on the project in 2021 as well. And I was making some changes, some necessary changes within the code. So if you see the long list, you would see that I was making those files way back in August 2021. In 2024 again, I was just going through the codes and writing explanations. Those explanations were note to myself. I should make it clear that I do not have anybody to thank this for, because at that point of time I was not visiting any organization and I just did this coding all by myself. The idea came into my mind, the coding that I did by myself. ClinVar now has this beautiful graphical representation where for a particular gene, say GLUT1 here, you can actually map the position of the mutations on the chromosome. Say for example here, at this particular location, 3 variants are reported. See, this red ones are pathogenic. Likely pathogenic is orange, likely benign green, benign is dark green, conflicting is gray, uncertain significance is yellow. So that is their color code. And at this particular location, there are 127 variants where the particular mutation has been reported to be of uncertain significance. Back in 2020 and 2021, when I was coding, these color demarcations on clinical classification showing the exact number of cases were not available. They added this feature later. And I must say that now, even without the codes that I wrote, this section of correlation becomes easier. If not this one. Thank you for watching up till here. During that 2020 lockdown period, I was suffering from some health issues, and also my computer, my laptop was giving me issues. I'm grateful to my mother who was taking care of the household back then, to the Python book that I bought for myself and the internet connection that I had. Thanks to the vendors who fetched us necessary items at our doorstep during the lockdown period. To the few friends and relatives who we were in contact with via phone, thanks to them for providing moral support. I'm grateful to the physician who treated me right in May 2020 after I had undergone a series of wrong treatments. Thankful to IT hardware personnel who tried to fix my old computer's hardware. Last but not the least, I'm thankful to myself for carrying on the work despite the odd situations. And above all, I'm thankful to the Almighty. I take this opportunity to request you to check my channel for other videos that could be of relevance and of use to you. If you like the video and if you like the work I'm doing, then please support me by subscribing to my channel and by sharing these videos with whoever might find this useful.